Today, we're diving into a landmark 1985 study that literally mapped our brain's chemistry.
How can you actually see where a chemical is working inside the human brain? Let's find out. Scientists really needed a map of where serotonin docks in the brain to understand its job. They used a super clever technique. Think of it like creating glowing keys to find specific locks.
Okay, first up, they zoomed in on one receptor type, 5-HT1C, which is in the choroid plexus. They wanted to see how well different drugs could elbow their way in and bind to these spots.
And here are the results. See those curves? The further left they are, the better the drug sticks. This table gives us the hard numbers. A higher PKD value, that means a much stronger bond. Okay, but is a pig brain a good stand-in for a human one? To find out, they plotted the data.
And wow, look at that. It's a nearly perfect correlation. Those data points almost form a straight line. All right, next, the team looked at another key player, the 5-HT2 receptor over in the cortex. They did the exact same kind of test, and here are the new binding curves for that receptor.
So now we can do a straight-up comparison. Rat, pig, and human 5-HT2 receptors. How do they stack up?
Now this is where it gets really interesting. Putting these two graphs side by side tells a story.
The bottom line, rat and pig receptors are similar to ours, but they're definitely not identical.
The data is great and all, but where are these receptors actually located? Let's see them. Time for the photos. This technique gives us a visual map of where those keys landed.
And boom, you can see the 5-HT1C sites are just packed into the choroid plexus.
In the hippocampus, crucial for memory, they show up in these really distinct layers. See the dark bands? But check this out. The 5-HT2 receptors, they completely light up the outer layers of the frontal cortex.
Okay, we've seen the charts and the photos, but why does any of this really matter?
Well, it gave us our first detailed map and showed huge differences between species for drug research. This is exactly why you can't just assume what works in animals will work perfectly in us.
This 1985 map was a massive leap. It just makes you wonder, what can our maps show us now?